You came back. So this is number 3 in our series. The third video, see, I make this look easy, it's not easy. The third video, there we go, is going to cover building proteins. Rough ER, smooth ER, and Golgi. So let's go ahead and get started. First one is something you've heard of before. I mentioned we were coming back. Ribosomes. Ribosomes are the tiniest part of the cell. They are minuscule, itsy bitsy. Okay? Guys, when you draw a ribosome, most of the time, we draw them as dots. They're that small. They're so small they only have one job. That's all they can handle. Ribosomes' one job is to make proteins. They're very good at it, but that's about it. Okay? Now there are 2 types of ribosomes. The 2 types are what's called free and bound. So there are free ribosomes and there are bound ribosomes. And of course, each of these have an additional job. We'll do the free ones first. Free ribosomes get their name because in the cell, isn't it a lovely cell? Here's the nucleus. In a cell, they just kind of drift around. They're loose, they float in the cytoplasm, they really have no place to be. Yay, they're just loose. The proteins they make are going to stay in the cell. So these little squiggles represent proteins, and just like the free ribosomes, they're not going anywhere. So free ribosomes make proteins that stay. Okay? So nothing too exciting here. Bound ribosomes are attached to the rough ER and to the nucleus. So that's what I mean by bound, is they're attached. Typically, I'll draw, for example, a rough ER and then cover it in dots. And so these dots are ribosomes that are bound to this rough ER. Okay? When they make proteins, the proteins go into the rough ER. There's a protein right there. From there, they're going to travel through all sorts of things we're going to talk about till finally they leave the cell. So that's what makes bound ribosomes unique. The proteins that they make leave the cell. The proteins for free ribosomes stay in the cell. Very important difference. Okay. So speaking of ER, there's 2 types. ER stands for, this mouthful, endoplasmic reticulum. Good news, I'll never require you to write endoplasmic reticulum. You can always just abbreviate it ER. 2 types, smooth ER and rough ER. Very basic difference. Rough ER is covered in ribosome. Look at the dots. Ribosomes make it rough. Smooth ER. No dots. No ribosomes. So it's nice and smooth. Okay? Pretty simple difference in how they look under the microscope. Now, of course, you know we're going to talk about function. The rough ER is, again, covered in ribosomes. So when you draw it in your cell book, just draw me something and then put dots all over it. That's going to make it nice and rough. Okay? The ribosomes make, what do the ribosomes make? Do y'all remember? You learned this. Ribosomes make proteins. So, of course, the rough ER, since it's covered in ribosomes, its job is to make proteins. All of the proteins made in the rough ER go into the middle of the rough ER. That's why we draw it as kind of like this hollow tube. Here on this little animation, I love how they say rough with ribosomes, these black dots you can kind of see, that's the proteins made by the ribosomes filling up the rough ER. Okay? So, do you think the smooth ER makes proteins? Can't. Doesn't have any ribosomes. It has to make something else. It's going to make lipids. It makes fat and oil because all it is membranes. Remember, membrane is lipids. So it just makes more of it. Okay? So on your analogy, think of what makes. If I was doing a sports team, which athlete makes all the points or makes the big plays? If I was doing a salon, who makes all the hairstyles? If I was doing, say, a mall, who makes all the goods or who makes the sales? Think of who makes in your analogy. Now, once the proteins are made in the ER, so over here, notice we've got our rough ER. I say rough because, oh, look, there's a ribosome. We're going to travel to the Golgi apparatus. Golgi apparatus is also sometimes called the Golgi body. Golgi body looks like, to me, it looks like a stack of pancakes. It's just a lot of membrane. I forgot how to spell pancakes here. Look at that. It's just a lot of membranes all piled one next to the other, next to the other, next to the other. Okay? Its job is it's kind of like the post office of the cell. It packages proteins that it gets from the ER, and then it ships them. So packaging and shipping. Proteins come in, they get modified somehow, things happen to them, and then they leave again. Okay. Here's a cute little animation. And just like I said, the proteins come in, they get modified. In this case, they get packaged. Oop, where'd it go? It'll come back. Packaged. Or you can say modified. And then they leave and get shipped off. So very, very much like the UPS of the cell. Okay, so here's a nice picture. This comes straight out of your book, and you can see all the parts we've talked about. The rough ER, making the proteins, traveling to the Golgi where they get modified, and then leaving, bye-bye, leaving the cell. Okay? Now, you're going to have to draw these for your book. You don't have to put them all on the same page. You can divvy it up. One page is rough ER, one page is smooth ER. Here in your notes, let's go ahead and do the whole thing. So this is going to be my cell membrane here. This is the goal I want to get to. I start at the rough ER. Rough ER is just kind of hollow. There we go. What's it missing? Well, it's rough. I got to add ribosome. So dot, 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 dot. Now it's a rough ER. Let's label it rough ER. The ribosomes on the rough ER make proteins. Squiggle, squiggle. The proteins are going to travel in vesicles. Squiggle, squiggle. Remember, vesicles were in video 2, I think. They're the cars, the transport of the cell. Eventually, one of those vesicles is going to travel to a Golgi. Golgi just looks like a stack of membranes all piled one on top of the other. That's enough. Golgi. Okay, the proteins are going to go in, squiggles. As they move through the Golgi, they're going to be modified in some way. This one's getting longer and longer. And then eventually they're going to leave, I need a bigger vacuole, and travel out of the cell. Now, I skipped one. I do apologize. Sometimes the things made by the Golgi don't leave. If it doesn't leave, it stays around, and it has all these proteins inside of it. And guys, that's where lysosomes come from. So lysosomes are vesicles made by the Golgi. This, by the way, is number 20 in your notes. I skipped it. But it's an organelle made by the Golgi for digestion in the cell. Okay, so that finishes off this video. That's 3 down. Not bad at all. We should be on the last page of the notes, next to last page, excuse me. And next up will be video number 4, covering energy. I'll see you there.